comes science. They might be giants, here comes science.
Hi, Dr. H here, and this will be the first video in my unit by unit review series, getting ready for the 2020 AP biology test. Today's video will be recovering Unit 1, which is the elements of life, and we will have 3 parts in this video. This first part will go over just some basics of chemistry. Part 2, we'll get into some of the biological molecules that we see in cells. And finally, part 3, we'll go over some of the important aspects of water and some associated concepts there. All right, so let's jump in here. First, we'll just look at a very quick overview of some of the elements. Certainly, you should remember that elements are arranged on the periodic table here by their atomic number, right, which is the number of protons. As biologists, we don't need to worry about all 118, I think it is now, of these elements, right? We can focus on just a few of them that we will see in living things, and those few are highlighted here. Certainly, the big 6, right? Carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. You should recognize those. And then probably the along with the ones that are highlighted in purple, they're the major ions, right? Things like sodium, potassium, chlorine. I think just recognizing these chemical symbols, right? The element symbols, should be enough, right? They're not really going to get into a lot of the atomic structure, atomic mass, number of protons, and that type of thing. Looking at moving on to atomic structure, remember that the nucleus of the atom is made up of protons and neutrons, right? Protons with a positive charge, and then the electrons, the negative particles, are found floating around in the energy level, right? In these electron cloud. Of course, in a neutral atom, the number of protons would be equal to the number of electrons, right? To cancel out the charges. When we think about these electrons in the energy levels, remember that these energy levels have a maximum capacity. For us, again, in biology, we don't need to worry about all the possible energy levels. It's really just the first 3 that we have to worry about. And those will hold 2 electrons in the first energy level, and then electrons will go into the second energy level, which can hold up to 8, and that is the same for the third energy level. Right? Again, I don't see them really asking, how many electrons are in boron or something like that, but just remember this 2-8-8 pattern, and you should be fine. The other important thing to remember with these energy levels is that for an atom to be stable, the outermost energy level, whichever level that is, needs to be full. So that is why elements and atoms will undergo chemical reactions and form chemical bonds to fill up that outermost energy level. So when we think about chemical bonds, that is the important aspect to remember, that this outer energy level needs to be filled. And when we look at chemical bonds, we can certainly kind of identify 2 major types of chemical bonds that we would see in biological molecules. Those are covalent bonds and ionic bonds. So the difference between these has to do with what happens to the electrons, right? Are the electrons shared between the 2 atoms, or are they completely transferred from one atom to another? And what determines that behavior is something called the electronegativity of the element. And you can think of electronegativity as the the strength that the atom, that the nucleus, right, the positive nucleus, is holding on to those outer shell electrons. Right? A highly electronegative atom is going to have a very strong pull on those electrons, and it can actually pull electrons from other atoms. A weakly electronegative atom is going to give up electrons fairly easily because it's not pulling on them very strongly. So looking at electronegativity values, the general trend is they are highest in the upper right-hand corner of the periodic table, and they get lower down to the lower left-hand corner. So thinking about biologically relevant elements, fluorine, which is the highest electronegativity atom that we see, not very commonly found in biological systems, so we don't need to worry about it. The right next to it, though, is oxygen. Right, oxygen is the most electronegative element that we will come across normally in biological systems. So whenever you see oxygen in a molecule, that should immediately tip you off that, okay, this is a highly electronegative element. It's going to be pulling those electrons. Okay, so what are these two main types of chemical bonds, and how are they formed? First, we'll talk about ionic bonds, and these generally form between two elements that are very different in electronegativity values. So that one atom, the highly electronegative atom, can actually strip that electron off of the lower electronegative atom and can ionize them. Right, so we see an example, sodium chloride, right, classic ionic compound here. Chlorine, very electronegative. Sodium, very weakly electronegative. So chlorine pulls that electron off. Sodium becomes positively charged because it gave up that negative electron. Chlorine becomes the negative ion because it gained that electron. And now the positive and negative ions are attracted to each other, and that forms the ionic bond. The other type of chemical bond are the covalent bonds. Okay, and these form when electrons are not transferred, are not moved from one element, one atom to another, but they're actually shared between two atoms. So these are elements that are closer together in electronegativity values. For example, here we have the covalent bond being formed between two hydrogen atoms. Right? Obviously, these will have the exact same electronegativity value. So these electrons are shared, so the electrons will now be orbiting around both nuclei, and that holds the atom together, right? These shared electrons. Now, when we think about covalent bonds, it's very important to remember that there are two different types of covalent bonds, and these are polar and nonpolar. And again, this has to do with the electronegativity difference, right? When two atoms are very close to each other in electronegativity values, then the electrons are equally shared, and we have what is called a nonpolar bond. Okay, so things like fats, a lot of organic molecules like methane here, these are nonpolar bonds where the electrons are evenly shared, and these tend to be non-soluble in water. The other type of covalent bond are polar bonds, and this is where the atoms have sort of this intermediate difference in electronegativities. You can probably look up tables to find exact values for the differences. I don't see that as being terribly important. But in this case with water, right, we have oxygen, which is fairly electronegative. Remember I said that was the most electronegative atom that we'll probably see. And then there's hydrogen, which is somewhat weakly electronegative. So oxygen is going to be pulling those electrons toward it, but not strong enough to actually strip those electrons off of the hydrogens. So the electrons are going to be shared unequally. They're going to be moving around the oxygen atom more so than they do around the hydrogen atoms. So that sets up this polar bond where this polar molecule, like water, has a partial negative charge on one end, the oxygen end, and partial positive charges on the other ends, the hydrogen ends. Okay, and that will have big effects on the chemical properties of these molecules. Okay, and we will see, as we go through and start to talk about the different biological molecules in part 2 of this video, we will see polar and non-polar bonds and how those will change the chemical properties of those molecules. One last thing to sort of talk about here very briefly, but before we get into the biological molecules, just some basic chemistry of carbon, right? This would be organic chemistry, right? Organic chemistry is the chemistry of carbon-containing molecules. So carbon has 4 electrons in its outermost shell, so it will form 4 covalent bonds to fill up that outer shell. Because remember, the third level, or sorry, it's the second level in carbon, needs 8 electrons. So those 4 covalent bonds will fulfill that 8 requirement. Carbon is very unique in terms of elements, in that it will form bonds with just about any other element around. Okay, it's not very picky about what it forms bonds with. So not only does it form these 4 bonds and make all of these really interesting shapes, but it can also form bonds with lots of other elements that we'll definitely see that when we get into in the next part of the video. So that will wrap up part 1. Part 2, like I said, we'll cover the biological molecules. Okay, so this is part 2 of our Unit 1 review, and in this section we will be going over the 4 major biological molecules that you should be aware of. First, we'll start with just some general information about these about biological molecules, and then we'll very briefly go into these 4 main types here. So to start, as I referenced at the end of the last video, or at the end of the last section, these biological molecules are primarily made up of carbon, right? And carbon, like I said, can form all sorts of interesting shapes, like chains, branch chains, rings. There are double bonds, triple bonds, all of these really cool, interesting things. And we'll certainly see some of those in our molecules. The other important aspect here is the chemistry of these molecules can change with the addition of certain functional groups, right? And these functional groups are just groups of atoms that have special chemical properties, right? And I certainly don't think you need to memorize the structures of these. Just kind of be aware of them. We will see them over and over again as we go over the 4 major molecules. And certainly the most important thing to remember, right, whenever we see oxygen, should always keep in mind that is a very electronegative atom and is going to give a little bit of a polar character to that molecule, right? Carbon and hydrogen together are highly nonpolar. Oxygen, with that, pulling the electrons away either from carbon or from hydrogen, is going to make the compound more polar. Okay, so most of these biological molecules that we're going to be looking at are what we call polymers, right? And a polymer is just a fancy name for a big molecule that's made up of repeating subunits, right? And these small subunits are termed monomers, right? Mono meaning one, and poly meaning many, right? So a polymer is just a lot of monomers stuck together, right? And we will see that, and I will definitely highlight all of the important monomers for each of the each of the major classes. So these polymers are built from a chemical reaction that's called a dehydration reaction. Right, so these here we see a reaction adding one more monomer to an existing chain. Right, so we're going from a polymer of 3 to a polymer of 4, and there is this reaction here between an OH that's a hydroxyl group and a hydrogen coming together. That makes a water molecule, right? H2O. So that is why this is referred to as a dehydration reaction, because water is coming out, right? And this new bond is formed, and now our polymer is one unit longer, right? One monomer longer. When the polymers are broken down into their monomers, then the exact opposite reaction takes place, where water is used to break the bond apart, right? And this is called a hydrolysis reaction, right? And hydrolysis literally means breaking with water, right? So water comes in, breaks that bond, and the monomer is released, and now the polymer is now one shorter, right? So this is what would be happening in your digestive system, as all of the biological molecules in your food are broken down into the building blocks to be absorbed. All right, so let's start talking about these 4 basic classes. All right, first we'll start with carbohydrates. So atoms that we see in carbohydrates, mainly carbon, hydrogen, and oxygen, right? We see a few examples here. I'm not going to get too wrapped up in talking about examples. For the most part, you don't need to memorize names, or certainly not structures of any specific molecules. Really just kind of understanding that the basic structure here, carbon, hydrogen, and oxygen, right? That's what we see in carbohydrates. Some general properties: very soluble in water for the most part, and certainly the smaller ones, the monosaccharides, those are the monomers, would be very sweet tasting. And we see the general formula there: carbon, a certain number of carbon atoms linked to that number of water molecules is the basic formula for a carbohydrate. Structurally, the monomer for carbohydrates is what's called a monosaccharide, right? One sugar, and then a couple of different polymers. If it's 2 monosaccharides stuck together, then that is a disaccharide, right? Di meaning 2, and then there are big, long chains of the monomers, and those are the polysaccharides. Monomer structures, they're most often in cells found in the ring form, as shown here in the diagram. They can exist in the linear form, but for the most part, once they're in a cell, they're going to take this ring form. The polymers, right, big long chains of these. We have one example here of a disaccharide, right? So this is sucrose, ordinary table sugar, and we see our dehydration reaction, right? The OH group and the hydrogen from the 2 different monomers coming together, forming the water molecule. The water molecule is released, and there is this bond that forms, this covalent bond that is holding the 2 monomers together, making the disaccharide. Polysaccharides, the long chains, form the same way, right? Just through repeating dehydration reactions, right? So here we see a couple abbreviated formulas for a few different polysaccharides like starch. That would be, these would each be hundreds of glucose molecules long, right? Glucose monomers. Some basic properties of carbohydrates. Monomers are used for the most part for quick energy. Right? Think like the simple sugars, really easy to digest. They're actually, they don't really need to do any digestion of the monosaccharides. They can just go right into your cells and immediately start providing energy. The polymers are more for longer term energy storage, right? Think starch, or there are also some structural features for these polymers, like the cellulose that is found in plant cell walls. Okay, so that's carbohydrates. The next class that we'll talk about are the lipids. Okay, and these molecules are unique in that they are the only one of these four biomolecules that are water insoluble. Okay, they do not mix with water. Structurally, or I guess not structurally, but the elements that are found, mostly carbon and hydrogen, right? That's why they're insoluble in water, right? They have all of these non-polar bonds. And there might be a few oxygens found in the mix, right? We see an example formula there, right? C16H32O2, right? So lots of carbon, lots of hydrogen, just a few oxygens. Lipids are also unique in that they don't really have a strict monomer-polymer structure, right? They are not repeating subunits of smaller building blocks as we see with the other three molecules. But we can still see these dehydration reactions forming the larger molecules. So, for example, one very common form of a lipid is a fat, right? A triglycerol, which means that there are three fatty acid molecules bound to one glycerol molecule, right? And as you see here, those are bound with a dehydration reaction, right? Oxygen, hydroxyl group, OH coming together, forming water. Water comes out and a covalent bond is formed, right? And we see the covalent bond highlighted there as the ester linkage. Structurally, in those fatty acids, right, those big long carbon hydrogen chains, there are a couple different properties that we can see. They can be saturated or unsaturated. Okay, and this has to do with the presence or absence of carbon-carbon double bonds in the tail. In a saturated fat, there on the left, right, looking at, for example, butter, that a saturated fat is all carbon-carbon single bonds, right, and it generally stays solid at room temperature, right, because the fats can all pack very tightly together. In an unsaturated fat, right, over on the left, like olive oil, there are these double bonds, right? So the carbon cannot rotate freely around that double bond, so the molecules have this little kink in it, right? That little bend. So the carbon, the fatty acid tails can't pack together so tightly. So this keeps these unsaturated fats liquid at room temperature, right? So things like vegetable oil or olive oil, as is in the picture there. A couple unique examples of lipids. First, we have the steroid molecules, right? This is cholesterol. Doesn't look like other lipid molecules, but it is classified as a lipid because it is insoluble in water, right? That is the most important kind of characteristic of these lipid molecules. And we'll see these again when we get into, we'll see cholesterol again when we get into membrane structure, right? There's a little bit of cholesterol in there, and a lot of hormones are also based on cholesterol. Another specialized example of lipids are the phospholipids that are found in cell membranes. Okay, and we will talk much more about these in the membrane review, which I believe is part of Unit 2. Okay, so I'm just going to show these right now, and we'll get into those in the Unit 2 review. General functions of lipids in the cell, because they are so water insoluble, they are really good at things like waterproofing, right? To keep surfaces, or to keep water in to an organism, or keep water out, whichever way you want to look at it. Insulation, right? Keeping heat in, they work very well. And a couple other things that I've already mentioned with the specialized functions. The hormones, right? Here are two cholesterol-based hormones. And then the phospholipids found in the cell membrane. Okay, and again, like I said, we will talk much more about those two as we get to those units. All right, so that kind of wraps up lipids. Our next molecule that we're going to talk about are proteins. Right, and this is probably the one that should probably be the most familiar with on this molecular level, right? Because they do really like to ask questions about protein structure and really get into some weird stuff. I don't know why they like to ask this so much. General properties. There's a lot of variability in terms of the elements that can be found. Certainly, they have carbon, hydrogen, oxygen, nitrogen. Right? Those are the big four. But with proteins, we can also see some sulfur. Right? This would be the only biological molecule that would contain sulfur. And occasionally you'll see some phosphorus, right? Due to some modifications of the protein structure. The overall structure of proteins is highly variable. Right? Every protein is going to have a unique structure, and one other thing that they really like to focus on is this relationship between structure and function. Right? You change the structure of a molecule, you're going to change its function. Right? And the function of that molecule is going to impact its structure. Right? It's going to drive evolution towards a particular structure. So this very, very tight interplay of structure function really kind of first shows up here with the proteins. So talking a little bit about general structure, the monomers for proteins are amino acids. We have some the structures here of the 20 common amino acids that are found in proteins. Again, certainly don't think you need to memorize any of these in terms of structures or names. It is, though, good to remember that they are grouped by the chemical properties of their side chain, right? Of what's called the R group, right? So this purple piece, let me zoom in on one here, right? That purple structure there that is highlighted in all of them, that is constant throughout all 20 amino acids. And then that highlighted region is what we call the R group, and they will have different chemical properties, right? So we see the top there. Those are some non-polar side chains, right? So those would be hydrophobic. The green ones in the middle there are polar, so those would be hydrophilic. And then the bottom pink and blue ones are charged, right? So those would also obviously be hydrophilic, but they would be acidic or basic. So these side chains are what give each amino acid its unique properties, and by putting them together in unique order is what gives each protein its unique properties, right? Its unique structure and function. Putting the amino acids together, again, not surprisingly, we see our dehydration reaction, right? OH group on one amino acid, hydrogen on the other amino acid, coming together, water comes out, and now these two amino acids are joined together by this covalent bond, right? Referred to as a peptide bond. Okay, we're going to see this dehydration reaction in every single molecule here. So in terms of structure of proteins, like I said, structure of proteins is highly variable, right? Every protein has a unique structure. But we can talk about like general structural properties. And when we talk about protein structure, there are 4 levels of protein structure, right? The first level here is the primary structure. Right? And the primary structure, the first level of structure, is just the sequence of amino acids, right? Which of the 20 amino acids are used in this protein, and what is the order, right? So these amino acids in this order, that is the primary structure, okay? The linear chain of amino acids. The next level of structure, the secondary structure, is the way that this chain of amino acids kind of bends and wraps around itself, right? And this is due to interactions between with the backbone atoms, right? The carbons and the hydrogens and the nitrogens that are all found in the backbone. And when we look at the secondary structure, there are 2 major secondary structural features that we'll find in proteins. And those are the alpha helix, it's on the top there, which is a spiral of amino acids. And then there are the beta sheets, which are sort of these flat sheets that sort of go up and down a little bit that form kind of flat areas of the protein. Okay, so those are the 2 secondary structural elements of proteins. The third level of structure now is what we call the tertiary structure. Okay, and these are interactions between the side chains, right? Between the R groups of the proteins. Okay, so this tertiary structure sort of gives the overall shape of the protein, and it's the way that these secondary structures, these alpha helix, I don't know why that says beta helix, that should say alpha helix, and beta sheets kind of interact with each other, and this is due to interactions between the side chains, or between these R groups. And there's a few different chemical bonds that can form, ionic bonds between positive and negative regions, hydrogen bonds, right, between polar regions, and there is actually a covalent bond. This disulfide bridge is a covalent bond that can form between amino acids that have sulfur, and actually really form a permanent piece of the protein structure. Okay, so those first 3 levels, right? Primary, secondary, tertiary. Many proteins only have those 3 levels, right? And that tertiary structure is the final functional version of the protein. But there are a handful of proteins that have a 4th level of structure, and that is referred to as the quaternary structure. And quaternary structure is multiple protein chains, multiple subunits coming together to form the final functional form of the protein. So here's an example of a quaternary structure. There are 2 separate protein chains here, right? And they both fold up with the first 3 levels. Actually, it says they're here. There's 4 chains. I guess if I'd actually read the slide, I would know that. These 4 protein chains come together and form this large functional transthyretin protein. Okay, and you need all 4 of these to be the functional protein. Okay, so all proteins have the first 3 levels, right? Primary, secondary, tertiary. A handful of proteins will have that 4th level, right? When there are multiple subunits that come in. One last important piece with protein structure is what happens when a protein loses its structure. Right? So structure function go hand in hand. So loss of structure equals loss of function. Okay, and this loss of protein structure is referred to as denaturation. Right? When a protein unfolds and loses its tertiary and secondary structure, right? And any quaternary structure if it is present. Okay, the primary structure generally stays, right? The amino acid chain, as you can see there, that amino acid chain nothing happened to that, right? The amino acids are still there in the same order, but the folding has gone away, right? The alpha helices have left. The tertiary structure, right? The way those alpha helices folded around each other, that is all gone, right? And this denaturing can happen when there are changes to the environment, right? Things like heat, extreme changes in pH can cause a protein to denature and lose its function. Okay, and this is a very important concept that will come up again moving forward as we talk about different protein functions as we move through. Now, in terms of protein function, it's really proteins pretty much do everything in the cell, right? So there's not one thing that we can point to that this is what proteins do. They do everything, right? And I think kind of have a diagram here showing some of their many functions. We'll talk about a few of these as we move through all of these unit by unit reviews. So it's really, there's not much point in kind of talking about each of these functions now. It's more important to get them as we go through. Okay, and that brings us to our final biological molecule, the nucleic acids. Right, and this is going to be a very quick review because when we get into the functions of these, which I believe is Unit 6, we'll talk a little bit more in depth about the structure, function, and all that stuff with the nucleic acids. For right now, just some general properties. They are slightly acidic. That's why they're called nucleic acids. And in terms of elements, right, carbon, hydrogen, oxygen, of course, and large amounts of nitrogen and phosphorus. So a lot more phosphorus than we see in any other molecule, right? I said proteins may have a little bit of phosphorus. Nucleic acids have a lot. Structure, I'm sure we've all heard the DNA structure, the double helix, RNA structure, right? That's always a single strand, though as we see in our transfer RNA molecule here, it can fold back on itself and have regions of base pairing, but it's certainly just one single strand, right? And like I said, we'll talk much more about this when we get into the actual DNA section of Unit 6. Monomers of nucleic acids are the nucleotides, right? And a nucleotide has 3 basic parts: the phosphate group, 5-carbon sugar, right, the pentose, and then one of the nitrogen containing bases, right? And those the sugars are different in DNA versus RNA. Right? DNA, of course, has deoxyribose. RNA has ribose. Right? The difference there is that one oxygen molecule, or oxygen atom, I should say. And then the nitrogenous bases, there are 5 common bases: cytosine, thymine, and uracil are the pyrimidines, and adenine and guanine are the purines. And again, don't memorize these unique structures, right? It's not important. We'll get into the important aspects of these bases once we get into DNA and RNA function. And the polymers of these DNA, RNA, they are repeating chains of those monomers, right, of the subunits, right, and they are formed by dehydration reactions, right. I don't show it here, because again, we'll get into it when we cover DNA a little bit more in depth, right. But there is the structure of DNA. You can see all of the repeating subunits, right. They don't actually show the atomic structures there, but that's fine at this point. And then finally, the function of the nucleic acids. DNA and RNA function together in the information flow of the cell. Right? DNA is the information storage molecule. Right? It stores the genetic information, and then RNA works to retrieve that information, send it out to the nucleus here, if it in a eukaryotic cell, to be translated into a protein, right? And RNA will also have a few other functions in getting that protein translated. Okay, so that is our quick wrap up here of our 4 important biological molecule classes. Part 3 will cover water and a few associated topics there, and then that will be the end of Unit 1. Okay, this is part 3 of our Unit 1 review video, and in this video we'll be going over some important properties of water. We'll go over water structure, some important of the emergent properties that are caused by this unique structure of water, and then we'll close out with a little bit about the pH scale, which is related to water structure and function. Okay, so water structure. Water is, of course, H2O, right? 2 hydrogen atoms covalently bound to 1 oxygen atom. Remember, oxygen is highly electronegative, so it's going to be pulling on those electrons, pulling them away from the hydrogen atom, right? Not strongly enough to ionize and form an ionic bond, but strongly enough that the electrons are going to spend more time around the oxygen atom than around the hydrogen atoms. So the oxygen atom then becomes slightly negative, right? That little squiggly symbol there, right? That is a lowercase delta. That indicates partial. Right? So there is a partial negative charge on the oxygen end of the water molecule, and there are partial positive charges on the hydrogen ends of the molecule, right? Because of the polar property of this covalent bond. These polar covalent bonds, because they have these partial positive and partial negative regions, are able to form hydrogen bonds, right? And these hydrogen bonds are very, very important, right? Everything that we talk about here in terms of the properties of water have to do with these formation of these hydrogen bonds. Right, so we see the hydrogen bonds there are indicated by the little dotted lines, where the partial negative region of one water molecule, the oxygen, is interacting with the partial positive charge of a different water molecule, right? At the hydrogen end of the molecule. So these hydrogen bonds, they are relatively weak, but they are very, very important. As you can see, water here is forming 4 hydrogen bonds with water in the surrounding neighborhood. Any polar molecule will be able to form these hydrogen bonds. Here we see an example of another polar molecule, ammonia, with nitrogen being more electronegative than the hydrogens. Water and ammonia will form hydrogen bonds between them. Right. So any polar molecule is able to form these hydrogen bonds because of the partial positive and partial negative regions of the molecules. So the important properties of water are all due to these hydrogen bonds forming. Okay, and these properties of water are what we call emergent properties. Okay, and this idea of emergent properties, it's not only with water that we see this idea in science. Basically, what emergent properties means is that when we get a collection of smaller pieces and put them together in a certain way, that new structure will have unique properties that we may not have predicted based on our understanding of these smaller building blocks. And we see these emergent properties at every single level here of biological complexity. Right? So if you pick any of these, going up in complexity, they're going to have new properties. Right? So if we pick, organisms, right, we can know exactly how this flower functions on an organism level, but once we get them all together in a population, that population has new properties, and it will behave in certain ways that if we only ever looked at individual organisms, we may not predict those properties. Right? And the same thing with going up another level to communities. Right? Once we start adding in different species, they're going to interact in certain ways that are unique. Okay, and water has a few very important emergent properties that are very important for life as we know it. And again, I'm showing this picture again. All of these emergent properties are due to these hydrogen bonds and due to the polar nature of this covalent bond found in the water molecule. Okay, so let's go over some of these emergent properties, and we're really going to focus on the ones that really impact life. Okay, the first is adhesion and cohesion. Right, so this is molecules kind of sticking to each other, right, through these hydrogen bonds. Very important for transporting water in plants. Okay, these very tall plants, this tree, need to move water from the roots all the way up to the top. Okay, this is due to, and this occurs because of the cohesion of water molecules hydrogen bonding to each other, and then the adhesion of the water molecules sticking to the insides of these very thin vessels inside of the plant. Okay, so the water molecule is basically form a long chain all the way from the leaves out into the atmosphere, all the way down into the groundwater. So when a molecule leaves the plant, right, when it evaporates out of the plant, it kind of pulls that chain all the way along, all the way up, and brings more water in. Okay, and that is cohesion and adhesion. Water also has a very high specific heat, right? Meaning that it can absorb a lot of energy before it changes temperature, right? And also, water will tend to hold on to that energy, to that heat energy, a lot longer than land. So, when we look at climates around large bodies of water, we will see that closer to the water, the temperatures are more moderate, and as you move away, temperatures will get much, much higher in the summer and also much colder in the winter. Here's an example of along the the southern region of California, where along the coast, it's somewhere between 72, 75 degrees. Moving inland a little bit, you see it goes up. It gets up into the hundreds. Right, same latitude, right, same amount of sunlight, but because there's a large body of water there, the Pacific Ocean, it's cooling down those coastal areas because the water is absorbing all of that heat energy and not changing temperature. When water freezes, the hydrogen bonds get a little bit longer, right? The water molecules move a little bit further apart. So water will, solid water, ice, will float, right? It floats on the top, right? I'm sure all of you have put ice cubes in a drink and seen the ice cubes float to the top, right? Probably never even thought about how that could be important. But for organisms that live in bodies of water that would freeze over the winter, this is very important because water, these lakes and ponds will freeze from the top down rather than the bottom up. So what happens, a layer of ice forms along the top, and underneath is the liquid water. That top layer of ice will act as an insulator and keep the liquid water underneath at a fairly constant temperature and keep it liquid all winter, so that things like the little shrimp here can survive, right? If ice were more dense than water, it would sink to the bottom and lakes and ponds would freeze solid from the bottom up and nothing could survive the winter, right? They would all just freeze solid and die. Here, since the water freezes from the top down, the water stays liquid underneath, and our little shrimp friend here can survive the winter very happily. Let's see, solventability, right? Water is a very, very good solvent, right? It will dissolve ionic compounds very well, and a lot of larger covalent compounds will also dissolve very nicely in water. Ionic compounds dissolve well because since water, remember, has partial negative regions and partial positive regions, water molecules can surround both the positive ion and the negative ion, right? The sodium and the chloride in this case. Okay? And we're breaking apart salt ion. When we're looking at covalent molecules, like a large protein here, what's happening here is on the surface of the protein, there are, again, these slightly partial positive and partial negative regions due to polar amino acids. So the water molecules will interact and can dissolve the protein. Now notice when a covalent molecule is dissolved, though, the bonds aren't breaking, right? Covalent bonds do not break when a protein like this is dissolved, right? When an ionic compound is dissolved, those ionic bonds do come apart, right? The sodium and the chloride ions are separated by the water. Okay, covalent compounds all stay together. Okay, so those are three of the very important emergent properties. There are a few more. Those are really the most important ones, I think, right? And the ones that are most likely to show up, right? And that's really what I'm trying to focus on here. Okay, so the last piece I want to talk about is pH, right? This is the measure of how acidic or basic a sample is. And this is related to water in that the basis of the pH scale is the ionization of water, right? So here we have two water molecules, right? We see the hydrogen bond in between them. Every so often, this hydrogen bond will sort of become strong enough to actually pull a hydrogen off of one of the water molecules and add it to the other. Okay, and this is ionization of water, and we end up with these two ions, right? The positive ion, the hydronium ion, or H3O plus, and the hydroxide ion, or OH negative, right? The balance between these two ions is what is measured by the pH scale. You will sometimes see the hydronium ion, H3O plus, referred to as only a single hydrogen, right? H plus, just one single hydrogen ion. It means the same thing. They are used somewhat interchangeably. I think this here with the hydronium H3O plus is a little bit more accurate, but it's certainly not wrong to have the H plus, right? And we will see that. So the balance between these two ions, right? And here we see an example of a situation where they're only using H plus. The balance between these two ions is what is measured on the pH scale. In a neutral solution of just pure water, the hydrogen and hydroxide ions will be equal, right? So that's neutral. If there are more hydroxide ions, more OH negative ions, then that solution is basic. If there are more hydrogen or hydronium ions, right, the H plus or H3O plus, then that solution it would be acidic. Okay, and the pH scale, of course, as I'm sure you all have learned in the past, runs from about 0 to 14, right, with right in the middle 7 being neutral, and that is pure water. Lower numbers are more acidic, higher numbers are more basic. Here's the pH scale with a few examples. Certainly see that the acidic solutions are more common in a biological sense, right? We see more, gastric juice, tomato juice, urine are all slightly acidic. Basic substances, our normal body pH for blood is slightly basic. I think it's like 7.4 or so. Inside the small intestine gets fairly basic, right? About 8.5. But then once we get up above 10, we see those really aren't biological molecules or biological compounds, right? The equation for pH, it is the negative log base 10 of the hydrogen ion concentration. Like every formula that we talk about in AP Biology, this will be given to you on the formula sheet, so there's no need to memorize it. I don't think you're going to actually have to use this formula. I haven't ever really seen this formula worked into a question. The one important thing, though, that you do have to recognize with this formula, because it is a log base 10 system, that means that for every change of 1 unit on the pH scale here, that reflects a 10-fold change in hydrogen ion concentration. Okay, so if we go from pH 5 to pH 4, right, that's 1 pH level more acidic. That is a 10-fold increase in the hydrogen ion concentration. Right, going 2 pH units, right, from like 5 to 3, that would be 100-fold more hydrogen ions. Okay, so that I think is the most important thing with this particular formula, right? The fact that each unit on the pH scale corresponds to a 10-fold change in hydrogen ion concentration, right? I don't think you're going to actually have to use this formula on the test. Okay, though I could be wrong. They could throw a curveball. We never know. All right, one final topic with regards to pH is this idea of buffers. Okay, buffers are compounds that will resist changes in pH. Right? They can either absorb extra hydrogen ions or they can release additional hydrogen ions depending on the conditions. This buffer system here, the carbon dioxide in water to form carbonic acid. This is a very common buffer system. It's actually found in our blood and keeps our blood pH to be fairly stable. Right? When there are additional hydrogen ions added to the blood, they will react with the bicarbonate ions and form more carbonic acid. If the pH of our blood gets too high, gets too basic, then the carbonic acid that's there will break down to form more bicarbonate and will release additional hydrogen ions to lower the pH back down. Okay, so these buffers are very important in living systems to sort of keep the pH within a fairly narrow range, right? It's one of the homeostatic mechanisms that exists in our bodies. All right, so that is the end of our Unit 1 review. I hope that all made sense, and good luck on your test.
Dropping science like Galileo dropped the orange.